This video is the Biofilm pre-lab. Biofilms are groups of microorganisms that are all living together with their secretions, so all the stuff that the cells release. It creates this like gooey matrix where all the cells are stuck together, and then they're also usually stuck to a surface. Shown here is a scanning electron microscope picture of that. You've got your individual bacteria cells, and then you can see all of like the gooey secretions. One cause of those gooey secretions is quorum sensing, which we heard about with Bonnie Bassler's TED Talk. To remind you, quorum sensing is how bacteria can talk to each other. Bacteria cells act differently when they're alone versus when they are in a group, and they do that through communication using small molecules, and we call that quorum sensing. Those small molecules are secretions, so they're stuff that's coming out of the bacteria cell. Those secretions then help to make up the biofilm, like the goo that keeps all of the cells stuck together. Here's some additional pictures of what that would look like. You've got your bacteria cells that first are living somewhat in solidarity, somewhat alone. They start to release a small amount of molecules, figure out who's near them, how many cells are near them. As there's then more and more bacteria cells, the amount of secretions then increase until you have like this three-dimensional matrix that is then a colony of bacteria cells and all of their secretions together. There's many examples of this that you've probably seen in your daily life. So biofilms are just like a layer of goo, including the microorganisms that created like that goo. So some quick examples shown here on the top left is a shower curtain with some probably mold, fungus, but also some bacterial growth. And that then creates a layer of like slime, and we would call that a biofilm. Shown on the right here, there's pictures of rocks inside of a creek. You can kind of get the gist from this slide that a lot of biofilms tend to form where it's moist, even more so where it's nice and warm, right? Living organisms like warm places that are wet. Shown here in the center is a picture of a toilet bowl. I know when I personally went to Florida for a month, Mr. Sable never went into our guest bathroom, and our toilet bowl in that guest bathroom looked much worse than this simply because it was still standing water that didn't get touched for like 4 weeks. So you get this growth of bacteria and other microorganisms that start to secrete and talk to each other in there, and that forms the biofilm. Any Nemo fans out there might recognize this. Inside of a fish tank, you get a lot of biofilm growth from microorganisms. And if you're a contact wearer, hopefully your case never looks quite as bad as this, kind of an extreme example. But again, you can see that there's like that layer of microorganisms and their secretions. Over the past 5 years, people in DC have spent $9 million trying to remove biofilms off of some of the national monuments. They call it like a black slime is taking over Washington, DC. And that's essentially bacteria that's growing on the buildings. And it's not as simple as just like spraying it off with a hose because it's so adhered to the building, and it's a three-dimensional colony of bacteria that's growing there. So $9 million is no cheap cost to remove that. In this particular lab, we're going to focus on biofilms on your teeth. Hopefully you guys go to the dentist regularly, but every time you go to the dentist, I feel like they spend the majority of the cleaning time scraping your teeth, or at least they do for me. I have a permanent retainer on the bottom of my teeth, and I feel like they spend most of their time scraping around that permanent retainer. So what they're removing from my mouth and what they're removing from your mouth when you go to the dentist are biofilms. We call them plaque or tartar. Usually we hear the dentist and commercials talk about plaque. So plaque is bacteria and all of their secretions. If you don't floss your teeth every day, I would highly recommend that you do so. I feel like I always thought the only purpose of flossing was to remove chunks of food from your teeth, but that's actually really totally not the main purpose of flossing. The main purpose of flossing is to remove bacteria that are living on your gum between your teeth. And so by flossing, you dislodge that bacteria and break up the biofilm so that less plaque can form. Shown here on the top left is a toothbrush bristle covered in microorganisms, and you can see the biofilm on that toothbrush bristle. So you've got on your teeth right now, even if you have good dental hygiene, you've got bacteria all over your teeth who are trying to form a biofilm, trying to talk to each other, trying to reproduce, eat any little sugar residue that's left in your mouth. So good dental hygiene is extremely important to prevent those biofilms from forming what we call or know as plaque. All right, so now we'll get into the lab. Fortunately, I have some pictures from years past of what this lab looks like, so we can go off of those. But in this lab, you're going to be using a 96 well plate. We use those a lot in this class. This is what one looks like, and you can see up here for scale purpose how small the plates are. It's called a 96 well plate because there are 96 wells or 96 holes inside of that plate. Having a lot of holes or wells in that plate allows us to do multiple replications of the same experiment. So in this particular lab, our goal is going to be to figure out whether or not mouthwash can prevent the formation of bacterial biofilms. In the top row of your plate, you will have 12 wells. So there's like 12 going across here. 12 wells that contain bacteria. On the bottom of the plate, we're going to use those 12 wells to put bacteria and the mouthwash. Now, anytime that you're using bacteria in the lab, you've got to give them food so that they can grow. So all of the wells that we use, all 24, will be given food. We call that food LB. It's named Luria broth after the person who came up with the formula of what combination of foods work best to feed the bacteria. But we need food in every well so that we give the bacteria a chance to reproduce. So you can then make a prediction about in which wells do you expect there to be the most amount of growth. So you've got some that contain bacteria and food, some that contain bacteria, mouthwash, and food. After we fill up our wells with bacteria, food, and some containing mouthwash, we will then let them sit for 3 nights overnight in an incubator. Bacteria and most microorganisms like to be nice and warm and cozy. So in the incubator, it's body temperature, it's 37 degrees Celsius. It's nice and warm in there. So we should see that the bacteria find a nice cozy home and they're swimming in their food. So they should then be reproducing like crazy. Or the mouthwash might be inhibiting some of that growth. Then at the conclusion of the 3 nights that we let them grow, we would dump out the plate of bacteria. So we would be trying to put all of the loose bacteria down the drain. To remind you, some of the bacteria will be inside biofilm colonies. So some of the bacteria will be stuck in their goo or in their matrix, which is their biofilm. Those bacteria would not become dislodged when we dump the plate. So we are just dumping any loose bacteria. Then, shown in this top left picture, we fill all of the wells that we used with a dye called crystal violet. The crystal violet will then bind to any bacteria and any biofilms that are stuck to the sides of the wells. So each well is a little hole, and there might be some biofilm growth on the side of it. So we then fill that plate with crystal violet, and that crystal violet will get stuck to the biofilm. We then rinse off all of the loose stain, and we end up seeing what looks like this. There is some staining in the plate, and what is stained is the biofilm. So a more purple color tells us that there has been more biofilm growth. We can quantify that data, so rather than just using my own opinion of, oh, this one looks more purple and this one looks less purple, we use a machine called a plate reader to quantify that growth, meaning to get numerical numbered data telling us how purple is that well. So to just kind of recap, more purple is indicative of more biofilm. We want that as a numerical numbered data, so we use the plate reader. The plate reader works like a spectrophotometer. If any of you have taken AP Chem, you may have used a spectrophotometer, but it works by using light. So we've got our 96 well plate. I'm going to show you just 2 wells here. Inside of those wells, there might be some biofilm, and that biofilm is now currently stained purple with the crystal violet. The way that the plate reader works is that it shoots beams of light through the bottom of the wells, and there is a detector up at top that can detect how much light was blocked by the sample. So if I have a greater amount of biofilm, like let's say this one on the left has more biofilm than the one on the right, I would expect to see a larger number, meaning that more of the light was blocked by the contents of that well. On the right, I have less purple, less biofilm, so my light would more easily be able to go and pass through that sample, so I would get a smaller number on the plate reader. So what that means is that if there's more purple and more biofilm, you will get a larger number on the plate reader. A small number on the plate reader tells us that there is a small amount of biofilm. To conclude this video, you are now ready to start looking at the actual lab. A few quick pointers on the actual lab. What you are given in the lab is 12 wells containing bacteria with no mouthwash. You are also given 12 wells containing bacteria and mouthwash. Your goal will be to figure out, does this brand of mouthwash inhibit bacterial biofilm growth for this particular species? When you look at all of the 12 wells of data, you are going to average them. So you will find an average for each of these. Your bar graph will then contain only the 2 averages, meaning your bar graph should contain only 2 bars. There will be 1 bar for bacteria with no mouthwash and 1 for bacteria with mouthwash. What you are graphing, what those numbers are, is average absorbance.